So here's the stuff I'm familiar with. Am I keeping going now?
Yes. Yep.
Okay. So here's some of the research that we've done on TPP in potatoes at Plant and Food. We were pretty lucky actually. We had spuds planted out at Cronin Road. We were getting ready to look at doing IPM in potatoes. We thought it would be quite straightforward. Obviously, potato tuber moth was going to be a challenge. And then TPP arrived. So we then sort of landed running and basically started 7 years of research in small plot trials at Pukekohe. We started firstly with early crop trials. We followed on with main crop trials. We assessed all the insect infestations, everything on untreated potatoes over the seasons, and unsprayed potatoes. And we came up with a subsample scouting technique. Obviously, you can't look at all of your potatoes and the whole plant, so we needed to kind of make it something practical and applied that growers could adopt readily. We also looked at the relationship between the beneficials that we were finding naturally residing in the potatoes in relationship to TPP. We also did lab studies to sort of validate, okay, we've got these things residing in the crop, these beneficials, what are they actually doing? So we did some lab studies to quantify the amounts of TPP that they were eating and which life stages of TPP they were consuming as well. We also ran some trials validating looking at the oils as alternatives to the standard insecticides, and picked a winner that we took forward. So here's my favourite stuff. It's the beneficial insects. So in Canterbury and Pukekohe, we identified all of the insects residing in potatoes and then the key beneficial insects residing in unsprayed potato crops. No choice testing was done at Lincoln to identify the best predators, and in Auckland we tested with choice and no choice. So for the Psyllid choice experiments, it was comparing when they were offered aphids and TPP and also just when they were offered TPP in petri dishes. So the key species we tested were the lacewing, small hoverfly, the Pacific damsel bug, 11-spotted ladybird, and large spotted ladybird beetle, which was work that we actually conducted for HAL in Australia when they were figuring out what they had there to predate on TPP should it arrive there. That's a lovely lacewing larva you can see there, and also the photo on the left is a pupa of a lacewing. So yeah, lacewings, lacewing larva, lacewing adult. So the larva and the adult of lacewing both predate on TPP. And then we have the slightly different but kind of similar looking hoverfly eggs. They usually are laid in a group. The larva there is very voracious on TPP. They can eat up to 40 nymphs a day. The adult doesn't predate, but obviously propagates the larva, which is a very effective biocontrol agent. So here's some more. We've got 11-spotted ladybird larva there at the top left and the adult on the right. They both predate on TPP. And they're seasonal too. So lacewing tend to be sort of spring leading into summer. Summer we've got the hoverfly, and then sometime late summer, sometimes the ladybirds show up, not always, and they also eat each other and other predators. So they're definitely a player, but they are less reliable as a predatory insect. Sort of late summer, we start getting the nabbids. They eat all life stages of TPP. They were one of the few that was interested in the egg of TPP. And then the unsung hero is probably the spider. We don't always pick them up when we're scouting because they're out at night. You'll see the webbings around plants, but they're definitely doing some work taking out some of our pest insects, including TPP. So this is a busy slide. One of the things we looked at was action thresholds as one of the IPM tools. After 3 years of weekly monitoring, we looked at trap catches and we tested different thresholds of when to activate spraying. We definitely came up with one for early potato crops in Pukekohe that actually, generally speaking, you didn't need to spray. You could actually get that crop through relying on the beneficial insects and conserving them by not spraying. The threshold that we came up with that came with the least zebra chip that was commercially acceptable was no more than 3 TPP per adult. This didn't stand alone. It wasn't just if you've got 3 TPP on your sticky trap or more, you should spray. It was in conjunction with other information we were gathering from scouting and so on. So another tool, which I've just kind of described as the action threshold. So it worked in Pukekohe. It didn't work so well in Canterbury. So you do really need to look at your approaches to controlling TPP in a regional way. Canterbury tried the 3 TPP threshold and it was too late. It was actually too late. So the zebra chip was actually showing up higher in the subsequent crops. So they were looking at predicting the first TPP in a crop, and they had weekly trapping data from a large number of tomato and potato crops from several regions, and this was combined with temperature and other data. So this didn't actually pan out, and even though it was a failure, it was still information that was really useful. At some point, TPP on the traps just increased exponentially. In the North Island, in subsequent trials, 980 degree days from the 1st of July actually worked quite closely with 3 TPP on a trap weekly, but in the South Island it was more like 712 degree days from the 1st of July, but when boxthorn was present, 600 degree days was advised. So that's quite a range of different degree days to look at: 980, 712, 600. So definitely necessary to go earlier in the Canterbury region on the degree day accumulation. So another IPM tool was looking at the use of selective insecticides. So you've got to have a range of tools. We trialled different insecticides at Pukekohe, including oils within different regimes. And JMS Stylet oil came out as the winner in the oils that we looked at. It's a colourless food grade mineral oil with a really high purity, which the purity is very key to its efficacy. So we got going with some trials in 2012 using the oil in relationship to other insecticides as well. And this was also part of a project that Peter Wright was involved in to support registration of JMS Stylet oil on potatoes. I don't think it's actually currently registered for use on potatoes per se. We also looked at different rotations of different mode of action groups, obviously to minimise build-up of resistance. Using too much of the same mode of action is a risky business. So we looked at having as many selective insecticides as possible in our armoury. We also looked at the impact of different insecticides and oils on beneficial insects, some of that in the lab, but also scouting, like, do you find as many beneficials on the oil trial areas? Another tool we had, and this was, I know, was on the Potatoes NZ website for growers to look at, was the degree day calculations. This was a mata one that I looked at every week when I was going in to scout the crops in there. Where are we sitting? So there is 3 years represented there. Time between generations obviously is shorter as the temperatures are higher. So here's a beautiful photo of a site in Matamata. This is an A.S. Wilcox and Sons site. This was looking at plantings of things such as Phacelia to enhance your natural enemy populations. This was a collaboration with A.S. Wilcox and Sons. We did 7 assessments at different sites in Matamata, but also in the Auckland region. What we found was that hoverfly numbers was really statistically significantly increased in comparison with the site that we scouted at the same time that did not have Phacelia. In Canterbury, they also did a kind of a similar look at grass strips planted alongside potatoes to see if that supported beneficial insects and, you know, ideally helping pest management. So the numbers of hoverflies at Phacelia sites range from 7 per middle leaf from 5 metres away from the Phacelia, and it drops off to just over 1 per middle leaf 100 metres away, but between 5 and 100, still a significant increase in hoverfly activity. We found it didn't make any difference to the lacewings. They were just there anyway. They didn't seem to increase in numbers with the Phacelia. But it definitely appears that Phacelia plantings are an important reservoir for hoverflies and therefore can be a useful tool towards an IPM program in potatoes. So agricultural oils were tested between 2011 and 2014 in the lab. We tested agricultural oils and mortality and Liberibacter transmission. So there was definitely repellency of TPP and increased nymph mortality compared with the water treatment. Fewer adults, the nymphs, the TPP nymphs failed to complete their life cycle, somewhat more in the oil treatments, and there was a slight reduction. I'm guessing it was not statistically significant, but there was a slight reduction in the transmission of Liberibacter as well. In Canterbury, XL oil and JMS were both tested. There was no foliar burning noted, and TPP control under low pressure was obtained with a slight reduction in the beneficial insects. So the oil in comparison to the untreated, there was a slight reduction in the beneficial. So the oil was slightly more impactful on the beneficial insects. So we took 7 years of research into commercial sites in collaboration with A.S. Wilcox and Sons. So we were basically taking some pretty sound strategies with us. We didn't want to go onto somebody's farm and have it blow up. So we felt reasonably confident that we had something that we tested and put through its paces. We were fortunate that we were able to take this to a larger scale because small plot trials have their limitations. What we took with us was scouting methods, sticky trap thresholds, it's the 3 TPP day a week, and degree day information and JMS oil with us. We're also really excited to take this to a different location. So we demonstrated it worked at Pukekohe. Could it work in another region with different soil, different climate? This is a question I asked Brian. I don't know if he remembers this, but why reduce insecticides? I'm not going to read that out, but the reasons, some of them seemed very clear to me, but obviously the environment's a key one, conservation of beneficial insects. It's an important thing for brand. It's nice for staff if they're not putting on insecticides every week. Being good neighbours was a nice one. So here we are. That's Matamata trials just gone in. We did 3 consecutive main growing seasons, and we asked A.S. Wilcox, to estimate the number that typically go on of insecticides in this region, and generally speaking, it was between 4 and 16. So how did we do it? We had a reduced spray area adjacent to the standard spray regime. In this graphic, on the left top edge is the standard spray regime. This particular planting went in October 2017. On the right-hand side, we have the reduced spray. So if you're looking at it as a racetrack, basically when we got to the end, the standard spray had 13 insecticides go on, and the reduced had 5. That was achieved by delaying the spray initiation, but also just by alternating the standard spray regime with an oil. So we were basically subbing out an insecticide every second week. So yeah, we scouted prior to initial sprays. We looked at degree day information, sticky trap information, and the alternation of the insecticides with an oil. So what happened? Obviously, there were significant insecticide reductions simply by halving the amount that went on. We assessed the crops, the yield, specific gravities, and zebra chip, and there was no statistical difference between the standard spray regime and the reduced. So that was between 2016 and 2019. So all the spuds that were in the reduced were combined with the standard and used for commercial purposes. Another IPM tool that sort of sat outside that particular project was you've got to have a good biocontrol agent. There's very few IPM systems that work without one. This was a very industry-led program of the importation of Tamarixia triose. We were asked to facilitate the importation of it, which we did into containment at Mount Albert in Auckland. And first of all, we conducted non-target hosts. So we had colonies of native psyllids that we tested against Tamarixia triose. There was attack in some of the native psyllids, but it wasn't considered significant enough to say, no, thank you, we don't want it in New Zealand. So the EPA decided that that was a go. We sort of developed our rearing system of Tamarixia on TPP, and then once it was cleared for being released, we transferred that colony and rearing system to a commercial biocontrol company, which was Bioforce, and they took it on. It was released officially using SFF funds between 2017 and 2018 at 2 sites in Canterbury, 3 sites in Hawke's Bay, and later in some covered crop systems in Auckland. It did establish in Canterbury and Hawke's Bay, but surveys of the regions of release in Auckland did not detect it, which I suspect is probably due to the use of insecticides. Most parasitoids don't really thrive in a setting where there's frequent spraying. So another IPM tool is developing sustainable spray programs. 2013, 2014 it was Pukekohe and Lincoln. 2014, 2015 Southbridge, Pukekohe and the Manawatu. 2015, 2016 in Southbridge, Matamata and the Manawatu. The aim was to reduce the number of synthetic insecticides in the spray program. We used a standard spray program and a no treatment control. Extended spray intervals were trialled, 7 days between sprays. This was using degree days and the 3 TPP per trap action threshold and alternating with Excel. Mesh was trialled in Southbridge, and the results were mixed. TPP should be TPP management, as I've said already, should be adjusted to the growing region. Oils can be a part of the spray program, but extended spray intervals were not successful. In this incident, the mesh size was too large, and you can imagine if the odd aphid got inside under that mesh and reproduced, that's trouble, and the beneficials don't really have as much access to dealing with the aphids, so that was not a go. So another IPM tool is future-proofing spray programs. So in 2018, 2019 in Canterbury, anticipating the losses of OPs and SPs, they tested against a standard Canterbury program from 100% emergence to a standard program alternating with Excel from 100% emergence, and the standard program from degree day action threshold, standard program alternating Excel oil from degree day action threshold. And there was no significant difference between tuber marketable weights or numbers. However, the zebra chip incidence was lowest in the future-proof spray program, but not significant to the other spray programs. So here's a biggie, is understanding the other host plants that TPP are hanging out on if you don't have spuds in the ground. So looking at this between 2012 and 2015, all TPP life stages were present on non-crop host plants throughout the year. So they are overwintering, hanging out on something else, another solanaceous plant. This included Lyceum and Convolvulaceae species, all from the solanaceous plant order. The low background population of TPP flying around in the environment. So when African boxthorn was present adjacent to a crop, there was increased activity nearby, and definitely an edge effect could be observed in the host crop. African boxthorn and the native poroporo are really suitable hosts for TPP, not just to hang out on, but to develop and cycle on. So another big body of work that I know Jessica was very keen is looking at resistance. So looking at insecticide susceptibility in TPP in Canterbury. This was 2021 to 2022. Tested selected contact and systemic insecticides, and compared the field collected TPP in Canterbury against a lab susceptible colony, so a lab colony that hasn't seen insecticides for quite some time. It was found that there was no material difference between the mortality of adults and nymphs between the field collected and the susceptible, so that was good news. In the systemic insecticides, the transition from the to be assessed life stage to the next life stage reflected that the label manufacturer's claim was reasonable. So fundamental research, how do TPP interact with each other, the environment and the host plant through sensory cues, that's smell, vision and sound, looked at in the lab but didn't work in the field extension. Needing to understand the TPP population genetic variation, like are these strains, are we dealing with different behavior in different regions? Going deep dive into this, the mitochondrial DNA showed large variation in genetics. I'm not even going to say that word. I'm going to try endosymbionts, so internal bacteria. Are there some resistant genes that we haven't picked up? And also understanding how host plants respond to TPP and Liberibacter to support our knowledge around breeding. Oh wow, I finished. There's some publications relating to some of the North Island trials. That's Matamata, I believe, that photo, on a slightly stormy day. And that's a heart-shaped potato that I found on the 21st of June about 10 years ago. That's me. Big thank you to all the New
Thank you,
Zealand
Frances.
growers. I was just going to
Jessica,
say
now-
thank you. Sorry. So I was just going to say thank you to all the growers who let us wander into their trials, into their crops and conduct our trials. Thanks to all PFR colleagues, agronomists, scouts, ag chem industry for their support and help. And a thank you to the Sustainable Farming Fund, Potatoes New Zealand, Canterbury Potato Liberibacter Initiative, Australian Plant Biosecurity Cooperative Research Centre and Plant and Food for the bulk of the funding of this research. Now
Thank you,
I'll shut up.
Frances. And Jessica, of course, has just arrived just as you finish. Good timing. We would like to ask the audience now if they'd like to ask any questions. If the people that are online would like to ask questions, could you please do it in the chat? Someone will pick up those questions and we'll read them out to Jessica and Frances. So if there are any questions from the audience?
That I can quickly present my slides, but then I'm pretty sure I've got some stories. Is that possible?
Absolutely. Absolutely. Yeah, no, that'd be great, Jessica. Yeah. Come ahead and do that now.
Frances will need to drive your slides, Jessica. That'll be the only thing. Frances will need to drive your slides.
You not explained it so that she does.
I don't have Jessica's presentation.
Okay, am I going back in time?
Yes, you are.
Where would you like me to?